Okay, welcome back. Sorry about the interruption to the last video on mutation. So I'm just going to do a little bit about sickle cell anemia. Now this is a very, very famous and on your syllabus example of a substitution mutation. It's caused by one base change which causes a change in an amino acid. Which affects the protein hemoglobin. So what happens to the protein when it is affected is that the allele, if you imagine your red blood cells are kind of these donutty shaped things, and at low oxygen in somebody with normal hemoglobin they're still donutty shaped, but in somebody with this abnormal gene they change into a sickle shape. If you don't know what a sickle is, can I just Google image it? It'll be fine. You'll be able to work out what it's for if you Google image it. So these cells are called sickled cells, hence we call it sickle cell anemia. Now what happens is that they will not fit down capillaries, and so in low oxygen concentrations, for example if you're exercising or you're up a mountain and your blood has a lower oxygen concentration, a lower partial pressure of oxygen than normal, any cells that have got this abnormal hemoglobin will sickle, and they block capillaries. And therefore they cause crippling joint pain and anemic symptoms where the oxygen's simply not getting through. Now the reason that this is very famous is it sort of fits into other things. So interestingly, the mutated allele, which I'm going to call S, is codominant with the normal one. So, you know, if you are mating, if you've got two heterozygotes, you're going to get that odd ratio, aren't you, where you've got HS crossed with HS, and you're going to get 1 to 2 to 1 of normal. Ones that are sort of half sickle cells and half not, which we call two sickles, we call that trait. So 2 with sickle cell trait. To 1 that has all of their cells capable of sickling, and they have full-blown sickle cell anemia. So, well, okay, and there are a number of genetic conditions why is this one so important, and the reason that it's so important is you'll come across it again when we do evolution. Because actually in certain areas of the world this sickle cell trait has a much higher incidence amongst certain populations. So if you look at a map of sort of malaria versus sickle cell trait, you'll see there's a high correlation between the two, and particularly in Africa. there's a lot of things online about sickle cell traits. There's sickle cell trait societies, because obviously with the slave trade taking from the malarial areas of Africa, a lot of African Americans are still carrying this gene. Probably some of them are quite unaware. So this is just a little bit from a past exam question, and it shows the incidence of the malaria parasite, which, as you know, is one of the biggest killers in the world still. And also this shows the percentage of heterozygotes in the population, the number of people who have got percentage of people with sickle cell trait. And you can see that round Lake Victoria we've got this very high incidence: 27, 36, 40, 28, 26, 27, 21, 20. In the mountainous areas round here, it's not a great advantage to have sickle cell if you're living in a more mountainous or hilly region. 0, 6, 0, 0, 0, 3, 1. So these are concentrated around the lakes. Now that's partly because malaria is transmitted by mosquitoes. Mosquitoes need water to breed. The young then, the pupae, hatch out and fly off and go and infect people with malaria. They don't mean to do it, bless their little hearts. Now, so why this high incidence where we've got malaria? So in evolutionary terms, anybody, you and me, normal haemoglobin, have a high risk of contracting malaria, which can be fatal. If you've got sickle cell trait, in that particular environment, so not necessarily if you're living in New York, but if you're living in a malarial area next to Lake Victoria and you've got the genotype HS, then you are much less likely. The Plasmodium parasite really doesn't like that sort of sickle blood. Less likely to contract malaria. And if you don't contract malaria, this is going to give you a survival advantage. And as we all know, if you survive, you can reproduce and you can pass on your alleles, so you can pass on that mutated allele and protect your offspring. If you have full-blown sickle cell anemia, that means that your genotype is you've got both of your alleles being for sickle cell. Your cells are really likely to sickle. And this is a huge problem because it blocks your capillaries, it causes these very painful episodes when you're running about or you climb a hill. Not only that, but your spleen tends to remove those sickle cells as being damaged, and because you've got so many there, you're taking them away faster than you're replacing them, and that's the thing that causes anemia. It places the spleen and the liver under enormous stress, and therefore you're more likely to suffer from jaundice. I have to say the outlook certainly in Africa is not good if you have full-blown sickle cell anemia. I think it's a lot better. We have more treatments in the Western world, and you can look those up on the internet. So what would seem to be really, this is a bad thing, we've got these sickle cells, actually in malarial countries is much better thing to have, much better than just having the normal haemoglobin, despite the sort of the downside of having, if you've got a particularly high incidence of heterozygotes in the population, they're more likely to have these matings where there's a 1 in 4 chance of having a child with sickle cell anemia who's probably realistically not long for this world. However, a 50% chance of having a child that is protected from malaria. So that's why sort of sickle cell anemia is quite important, and it's a really good example of a substitution. So the amino acid change is something like glutamic acid to valine. You don't need to know that. If they want to ask about it, they'll give you the base sequence. They'll probably give you one of these tables, or at least a section of it, so that you can work out what that mutation actually is and what the base change is. What you need to know is the effect on the protein. So it's causing this different protein which sickles the cells, and you need to know its advantages. It's quite a nice one to be asking, because it covers a number of different syllabus areas, obviously, and that's always a bonus for an examiner. Anyway, good luck with